So we'll start our discussion of the cell-mediated immune response with T cell activation. T cells can only recognize antigens that are shown or presented to them in a special membrane receptor. These membrane receptors that can perform antigen presentation to T cells are called major histocompatibility complex proteins, or MHC. We have two classes of MHC, class 1 and class 2. Class 1 MHC receptors can be found on all the nucleated cells of your body, and it plays a role in the tolerance, that self-tolerance that I mentioned, since they normally present self-antigens. However, if that cell is infected with a virus, the surface receptor will present viral antigens. The T cells will recognize these viral antigens as non-self and destroy the cell. That'll stop the viral infection from spreading. Class 2 MHC proteins, these receptors are found only on the surface of lymphocytes and special antigen-presenting cells called APCs. And these antigen-presenting cells engulf and break down pathogens or foreign antigens by phagocytosis, and fragments of the foreign antigens are then displayed on the cell surface in this class 2 MHC protein, these receptors. And that's specifically for T cells to respond to. And I've used the term recognize, but T cells, remember, they don't have eyes like we do, but they do have their own set of cell surface receptors that act like their eyes in this case. They're called cluster of differentiation, or CD markers. There's two sets of CD markers that T cells can have, and that gives them the ability to respond to the two different classes of MHC proteins. Those T cells with the CD8 receptor specifically monitor the class 1 MHC proteins, and those T cells with the CD4 receptor monitor the class 2 MHC proteins. So depending on which CD marker the T cell has, and therefore which MHC protein stimulates it, determines the T cell's response. Based on their response, T cells are subgrouped into cytotoxic T cells, helper T cells, memory T cells, or suppressor T cells. The big hitters of the cell-mediated immunity are those T cells with the CD8 marker. Upon stimulation by antigen presentation in a MHC class 1 surface protein, these cells will become activated cytotoxic T cells and memory cells. The cytotoxic T cells will give the cell that's presenting the antigen in its MHC class 1 cell receptor the kiss of death, meaning that the cytotoxic T cells live up to their name. They secrete lymphotoxins, which will disrupt the target cell's metabolism. They secrete cytokines that activate apoptosis, or genetically programmed cell death. And they secrete perforins that rupture the plasma membrane. So here you can see antigen recognition and activation of a cytotoxic T cell. So a lot more detail than that earlier figure I showed you. Here we have an infected self cell that has been infected with either a virus or a bacterial antigen. And that is being complexed with that infected cell's MHC class 1 protein on the surface of the cell. You can see the little red antigen pictured there. And then where you see this T cell receptor, that's your MHC class 1 surface protein that activates that T cell. And that activated T cell becomes a cytotoxic T cell. It also produces some memory T cells as well. The cytotoxic T cell secretes lymphotoxins, which disrupt the target cell's metabolism. The cytokines activate apoptosis, and perforins rupture the plasma membrane. So this infected cell becomes lysed and broken down. It dies. Okay? Which is better. It's better to kill 1 or 2 infected cells, even if they're your own cells, because you can stop the viral infection there. The other major group of T cells are the helper T cells, which all share the CD4 marker. These cells are like orchestra conductors, meaning they coordinate specific and non-specific defenses with their cytokines, playing a key role in both the cell-mediated and the antibody-mediated immunities. So upon recognition of an antigen bound to an MHC class II protein on an antigen-presenting cell, the helper T cell divides and produces more active helper T cells and memory cells. And I'll tell you more about those cytokines in the next section. So as I mentioned, both the cytotoxic and helper T cells can divide into a memory cell population. So at the time of their creation, these memory cells are inactive, and that's because they're going to persevere through the current infection, and they're held in reserve for any possible subsequent exposures to that same identical antigen. In the event that that same antigen is encountered again, these memory cells are primed and ready. So their response will be so swift and efficient that the individual usually does not suffer from any signs and symptoms of the pathogen. Suppressor T cells are a special population of CD8 cells that develop very, very slowly. Often they don't become fully activated until the end of the immune response. Their role is to dampen the response of other T cells and B cells by secreting cytokines that are called suppression factors. So these cytokines put the brakes on the immune response once that threat has been neutralized. So we're at another checkpoint. Go ahead and pause the recording and test your knowledge. So throughout this chapter, I've been telling you that B cells make antibodies. So let's discuss the steps involved in this process. B cells have a cell surface receptor that's a lot like the T cell's CD marker. However, the B cell receptor looks and acts just like an antibody that's anchored to the cell membrane. And remember, antibodies are specific, so they only bind to one antigen. Unlike T cells, B cells can recognize free antigens with their antibody-like receptor. And the antigen binds to that B cell receptor and triggers an endocytosis event that will process the antigen. That antigen is then displayed on the surface of the B cell in its MHC class II cell surface protein. The B cell is now sensitized. Once a helper T cell encounters this B cell and recognizes the MHC class II bound antigen, it'll secrete the necessary cytokines to lead to that B cell activation. An activated B cell will divide repeatedly, producing many daughter cells with the identical B cell receptor, and many of these cells will differentiate into plasma cells, which are essentially antibody factories. Some of these activated B cells will be saved as memory cells to provide a faster, stronger, more efficient secondary response if that same antigen is ever encountered again. And so here we have the steps involved in a B cell's response to antigen exposure. Notice how the surface of this B cell has both the class II MHC proteins on it and antibodies, or the B cell receptors. These antigens are specific. They bind to the antibody molecules, that B cell receptor, which then is engulfed and taken into the cell. And you can see now that that antigen is being presented in the MHC class II cell surface marker. And that B cell will float around until it finds the appropriate T cell. Okay, and the T cell will recognize that class II MHC with the antigen bound to it. Okay, and the T cell will then provide the necessary cytokine stimulation to activate that B cell. Okay, so when the B cell is activated by the T helper cell, by that CD4 T helper cell, then the B cell will replicate, make many daughter cells that are identical. Some of those daughter cells become memory cells, some of them become plasma cells. And those plasma cells, their only goal in life is to make antibodies that are exactly like that B cell receptor that was first bound to antigen. So let's take a closer look at those antibodies. The antibody structure is a Y-shaped protein with two parallel pairs of protein chains. One pair of long, heavy chains make up the constant segment and provide a base for the antibody. It has regions for binding complement and macrophages, or being bound to the surface of the B cell and acting as a B cell receptor. The antibody also has a pair of light chains with a variable segment at the tips. You'll see antigen binding sites labeled in this figure of the antibody, and these are the free tips or the fingers of the variable segments. This is what will determine the specificity of that antibody. On the right, we have a pathogen pictured. Notice how the antibodies bind to portions of the antigen called antigenic determinant sites. So it depends on a three-dimensional fit between the variable segments and the antigen. And so now you can see that it's forming an antigen-antibody complex. Most environmental antigens have multiple antigenic determinant sites. Entire microorganisms could have thousands of antigenic determinant sites. So antibodies are also called immunoglobulins, or abbreviated IG. And we have a number of different classes of immunoglobulins. IgG makes up the largest percentage of the antibody population and therefore is the most diverse class. It's responsible for resistance against viruses and bacteria, and it is so small that it can cross the placenta and provide passive immunity for a growing fetus. IgM, the next one pictured there, is a pentamer arrangement of IgG, like heavy and light chains. They're the first class of antibodies to be secreted, and they mainly attack bacteria. This is the class that's responsible for cross reactions of blood types. IgA in the middle is a dimer of two IgGs, like monomers bound at their constant regions. IgA is the most commonly found antibody in exocrine secretions like mucus, tears, and saliva. Therefore, this class of antibodies attack antigens before they enter the body. IgD is the B cell receptor, so it remains attached to B cells. And the last one, IgE, stimulates basophils and mast cells. So it plays an important role in the inflammatory response, which is number 6 on this slide. So we have many functions for antibodies besides just stimulation of inflammation. I've been talking about some of these already. So let's go through each one. Neutralization. Antibodies and antigen complexes can prevent antigens from attaching to your own human cells. So that threat has been neutralized. Agglutination and precipitation. The next one there. Antibodies can bind to several antigens to form large complexes, and this is a process called agglutination. You saw this when we did the blood typing. Precipitation occurs when these large complexes are so big and so heavy that they actually settle out of solution. Activation of complement, number 3. So when antibodies bind to antigen, it changes the shape and it allows it to bind with complement proteins and start that complement cascade. Antibodies attract phagocytes, like eosinophils, neutrophils, and macrophages, kind of acting like a red flag. And they enhance phagocytosis by creating a coating of antibodies and complement, which makes pathogens easier to phagocytize. This is referred to as opsins, or a process of opsonization. And then last, antibodies stimulate basophils and mast cells, which can mobilize a lot more of the nonspecific defenses like inflammation. So throughout these last few sections of the chapter, I've mentioned the memory T cells and memory B cells. The immune response that generates or creates those memory cells is the primary response. And this primary response takes time. The right B cells with the right B cell receptor has to encounter the right antigen, which then has to encounter the right T cell. And then those B cells need time to divide and differentiate into plasma cells. And so those plasma cells need time to generate antibodies. And during this time, which is about 1 to 2 weeks, the individual will start to experience signs and symptoms of the infection or the disease. Now, after that antigen's been neutralized, The memory cells, which were inactive during the primary response, now have the numbers. Okay, so the likelihood of the right B cell encountering the right T cell and antigen are much higher upon that second exposure to that same antigen. All of the memory cells will respond instead of just one cell. And the plasma will just be flooded with antibodies. More antibodies and sooner. The individual undergoing a secondary immune response is typically completely unaware that the infection is even occurring. And this can be observed in the titer, or the concentration of antibodies in the serum. So on the left-hand side, you can see there's an arrow that says antibody concentration in the blood, and on the bottom, you can see an arrow that says time in weeks. So the first exposure to an antibody, you have to go through all those steps that I just mentioned. The antigen has to encounter the right B cell, which has to then encounter the right T cell, which then has to activate that B cell, which then has to divide and differentiate into plasma cells, which then have to make antibodies. So all that time takes 1 to 2 weeks before you hit a peak of antibody concentration in the blood. Okay? And then after that threat's been neutralized, the B cells will stop making antibodies. I mean, you're always going to have a small amount of antibodies for that specific antigen floating around in the blood, but not the peak that was necessary to neutralize the threat. Upon second exposure, the memory cells, you have a ton more memory cells that are ready to go. So as soon as they encounter that second exposure to the antigen, they start cranking out the antibodies right away. And so you see this huge increase in the amount of antibodies in the blood that are specifically going to fight that antigen. Okay? And this primary response and secondary response, this is the idea behind vaccination. When you're vaccinated with something, that's your first exposure. You experience a primary response without having to experience the disease. So that if you ever do encounter that pathogen that you've been vaccinated for, you'll have a secondary response, and you might not even realize you were exposed to that measles or that mumps or that rubella, because your secondary response neutralized the threat before you felt any signs or symptoms. So this figure shows a real broad summary of the immune response and its relationship with the innate nonspecific defenses, because they are interwoven. They all have the same goal, the destruction of the antigens. But at first, you have antigens, which will trigger your innate nonspecific defenses, like the complement system and natural killers and macrophages. Some of those cells are antigen-presenting cells, and they will show those antigens to the members of your adaptive specific defense. For example, CD8 cells, if it's an antigen that's presented in that MHC class 1, or if it's a MHC class 2, that's going to be triggering your CD4 cells. Okay? Either way, you're going to have those T cells responding. If it's a CD8 cell, they're going to attack and destroy any abnormal cells that are presenting that antigen and destroy the antigen. You'll have memory cell population that's generated and lays in wait for a secondary exposure. And those CD8 cells have to differentiate also into a population of suppressor T cells, which will help the CD8 cells, the cytotoxic T cells, and any activated helper T cells. If we look over at the helper T cells, which are stimulated by that MHC class 1, will then differentiate into memory cells and helper cells and become activated. Those activated cells are going to be secreting cytokines to help the B cells produce and differentiate plasma cells, which make antibodies, and the production of memory B cells. And again, all of that is ultimately designed to destroy antigens and keep the body safe, provide immunity. So the body's immune response is coordinated by physical interactions, that antigen presentation, and by the release of chemical messengers or cytokines. Cytokines are often classified by the cells that release them, but not always. There's 20 different known interleukins, a name referring to between white blood cells, inter meaning between. Anyways, and with each different interleukin, there's a different function. So some of the better understand functions increase T cell sensitivity to antigens, they stimulate B cell activity and antibody production, they enhance the nonspecific defenses, and some suppress immune function. And I already discussed interferons, but as a review, interferons can make cells resistant to viral infections, and they can attract and stimulate natural killer cells. Sometimes, and we're doing research to see if we can use them to treat some forms of cancer as well. Additionally, we have tumor necrosis factors as a hormone of the immune system. These are specifically designed to slow tumor growth and try and kill those tumor cells before they become disruptive. So these are aimed to stimulate neutrophils, eosinophils, and basophils. Phagocytic regulators are going to be responsible to coordinate specific and non-specific defenses by adjusting phagocytic activity. And lastly, colony-stimulating factors are going to stimulate the production of blood cells in the bone marrow. And so we're going to be making more lymphocytes in the lymphoid tissue. So just making more colonies, more populations of lymphocytes. And so that does bring us to the conclusion of this lecture on Chapter 14.